all for being here. I've had a wonderful visit so far, and I'm really excited to tell you a little bit about my dissertation work on developing strategies for improving diagnosis of malaria and schistosomiasis in low-resource settings. So, as many of you know, the burden of malaria around the world is immense. In 2016 alone, the World Health Organization estimates that there were 216 million cases of malaria and 445 deaths due to the disease, the majority of which occur in children under 5 years old and in sub-Saharan Africa. While this burden is very troubling, there is some good news, and I always like to think about the good news. So at the turn of the millennium, the United Nations set forth the Millennium Development Goal of halting and beginning to reverse the rising incidence of malaria by 2015. And in those 15 years, the malaria community was successful in achieving that goal. The rate of new malaria cases decreased by 37%, and deaths due to the disease, the death rate due to the disease decreased by half. And that success can be visualized in the map on the right, which shows the percent of total deaths due to malaria in children under 5 in 2000 and 2015. And you can see that that burden was immensely reduced. And because of this progress, the World Health Organization in 2016 set an ambitious new goal of decreasing malaria incidence and death rate by 90% by the year 2030. And some of the most important tools for reducing the burden of malaria are diagnostics. And diagnostics are important in several contexts or use case scenarios. The first is case management. So if a patient presents to a clinic with symptoms, having a good diagnostic test allows the caregiver to determine whether or not those symptoms are due to malaria and then select the appropriate treatment. It also helps avoid inappropriate treatment, which can lead to drug resistance. For intervention management, having good diagnostics is important for evaluating whether an intervention such as distribution of insecticide-treated bed nets has been effective at reducing transmission within the population of interest. And for surveillance, having good tools to measure the prevalence of disease is important for identifying transmission hotspots and performing large-scale studies on a national level. And finally, what's going to be the focus of my talk, a lot of my talk, is the detection of low-density malaria infections. So having sensitive and specific diagnostics is important for detecting these infections, which many of which are asymptomatic. So a patient doesn't feel any symptoms and therefore doesn't seek treatment unknowingly and continues to contribute to the transmission reservoir. So I'm going to talk a little bit more about these infections. So symptomatic malaria patients typically present with malaria parasite densities on the order of 100 to 1 million parasites per microliter of whole blood. In contrast, asymptomatic patients have much lower parasite densities circulating in their blood system. So this is a study, in a study recently from Cambodia, Thailand, and Myanmar, they found that the average parasite density in asymptomatic patients was 5 parasites per microliter. But infections as low as 20 parasites per milliliter have been detected. And while it's still a topic of ongoing research, it's thought that patients with these low parasite densities still contribute to the transmission reservoir and can therefore transmit the disease to a mosquito to perpetuate the transmission cycle. And depending on the setting, these asymptomatic infections can represent anywhere from 20 to 80% of the transmission reservoir. So it's really important that we can detect and treat these infections. And so what are the tools that we have right now that are capable of detecting malaria? I won't go into too much detail on this slide, but the first two techniques, PCR and ELISA, are laboratory-based techniques that cannot be done at the point of care. PCR is our most sensitive method for detecting parasites, and it detects parasite genetic material. ELISAs detect protein biomarkers for malaria and other diseases, and one of the qualities that I just want to point out right now that will become important later in my talk is that ELISAs can take a long time to perform, anywhere from 3 to 8 hours, so it can take a better part of a workday to perform ELISA. Light microscopy is the gold standard for malaria detection, and rapid diagnostic tests have drastically changed the way malaria is diagnosed in the field. In 2016 alone, there were 312 million rapid diagnostic tests that were distributed throughout the world, and they represented a majority of the malaria testing done. These rapid diagnostic tests, or RDTs, also, like ELISAs, detect protein biomarkers, and they're great because they're inexpensive, they don't take very long to perform, and anyone can perform an RDT. Their primary disadvantage is that they're not sensitive enough to detect those low-level asymptomatic infections. In fact, recent models estimate that current malaria RDTs can really only detect anywhere from 40 to 60% of the infectious reservoir in certain settings. But models have shown that if we can improve the limits of detection by 100-fold, we would be able to detect 95% of the infectious reservoir, which would greatly benefit elimination campaigns. And so this brings me to the first key question of my work, which is, what are the clinically relevant protein biomarker concentrations that we need to be able to detect on those RDTs in order to find those low-density malaria infections? This question will be the framework for the first part of my talk, in which I'll tell you a story about an assay that I developed for detecting the 2 primary protein biomarkers for malaria. And the way I approached this problem was to break the conventional diagnostic format into its individual component parts. So I'll take you through the biomarkers of interest, how we selected the molecular recognition elements capable of capturing and detecting those biomarkers, and how we optimized the assay for maximum signal output, and put all of those components together to make a sensitive detection method for protein biomarkers. So the 2 primary protein biomarkers for malaria are Plasmodium lactate dehydrogenase, or plDH, and Plasmodium falciparum histidine-rich protein 2, or HRP2. plDH is produced by all 5 species of malaria, and it's malaria known to infect humans, and it's fairly conserved across all of those species. It's the terminal enzyme in the parasite anaerobic glycolytic pathway, so it converts pyruvate to lactate at the end of glycolysis during the red blood cell cycle, and it's an essential protein for parasite survival. It's thought to be natively present as a tetramer, and it clears from host circulation within days of parasite clearance. In contrast, HRP2 is produced only by one of the species of malaria parasites known to infect humans, Plasmodium falciparum. We don't know what HRP2 does in the parasite, but it has a really interesting primary amino acid sequence that consists of a series of histidine-rich repeats, hence the name histidine-rich protein 2. It's thought to form a dimer in the presence of heme, and it's known to persist in circulation for weeks after parasite clearance. And so we're interested in developing a tool that can detect both of these biomarkers. And there are several advantages for detecting both biomarkers. Of course, we're interested in determining the biomarker concentrations in low-density infections, but even from a case management perspective, having both of these biomarkers is really useful. So I'll just walk you through why this is useful. So the plDH portion of the test lets you know whether someone has an active malaria infection of any species. And then the HRP2 portion of the test determines whether that infection is due to falciparum or non-falciparum malaria, which can then inform treatment. Also, having the plDH portion of the test helps us avoid false positives due to persistent HRP2 remaining in circulation after an infection is cleared. And so, as I mentioned before, my objective was to develop a tool for quantifying plDH and HRP2 in samples with low parasite densities. So what are the qualities of an assay that we would want in order to be able to do this? Well, we're looking to have detection limits on the order of single parasite per microliters. We want efficient sample processing. So as I mentioned before, ELISAs can take anywhere from 3 to 8 hours, and we'd like to be able to get our data faster than that. We'd like to measure both biomarkers from a single sample, so have a high information output. And of course, we'd like our assay to be highly reproducible with low inter and intra-assay variability. So the way we approached this problem was a colleague and I independently developed and optimized individual assays for each biomarker. We then put these together into a dual detection multiplex test for both biomarkers. And the format that we decided to use was what's called an on-beat ELISA. And the way an on-beat ELISA works is we have magnetic particles that are coated with antibodies that specifically target our biomarker of interest. We also have detection antibodies that target that biomarker that are conjugated to an enzyme which can produce a signal when in the presence of its substrate. And so we add both of these components directly to a lysed whole blood sample. And if the target is present, we form sandwich complexes on the surface of the magnetic particles. We can then use an external handheld magnet to isolate those particles and remove any interference from the whole blood mixture before washing and probing for the presence of the target using the detection enzyme substrate. And there are several advantages of this on-beat format compared to the traditional well-plate ELISA format. First is the all-in-one format. So unlike ELISAs, which are stepwise and therefore take a very long time, we're putting all of our components for the assay directly into the sample. The magnetic beads also afford us a high surface area for binding, and we can actively mix those magnetic beads within the sample, which allows for much more rapid reaction times compared to traditional well-plate ELISA. And of course, we were very interested in magnetic beads because they afford us multiplex design capabilities, and we would like to use this to detect 2 biomarkers. And so the first step to developing an assay is to select the molecular recognition elements for capturing and detecting your biomarker of interest. So I'm going to tell you how I did this for plDH. And for plDH, we want a pair of antibodies that's capable of detecting plDH regardless of the species. In this case, I tested the antibodies against Plasmodium falciparum and Plasmodium vivax. And in order to do this, I performed 2 checkerboard plate ELISAs with 64 different antibody pairs against native P. falciparum LDH and recombinant P. vivax LDH. And these figures here on the right are heat maps of the relative signal-to-noise ratios that we got for each of the 64 antibody pairs. And of course, we want a pair that works well and equally well for both of these species. And so in order to figure out which pair was the best, I plotted the normalized signal-to-noise ratio for falciparum LDH on the x-axis and for vivax LDH on the y-axis. And each of these points represents a different antibody pair on these checkerboards. So an antibody pair which performs well would reside up here in the upper right-hand quadrant, and a pair that performs equally well for both biomarkers would reside on the line of identity. So you can see that we have 2 candidate antibody pairs of interest, and after further evaluation in an on-beat format, we found that this pair was the best pair for detection of Plasmodium lactate dehydrogenase. And so once we had our molecular recognition elements, the next step was to optimize the assay. I don't have time to take you through all of the components that were optimized, but I'll just show you a few examples of what the data looked like. So some of the things that we had to optimize were how many beads we added to the samples in order to capture all of the biomarkers. We also looked at the concentration of detection antibodies we added to the samples, the amount of time we incubated those components in the lysed whole blood samples, and finally how long we incubated the particles in the detection enzyme substrates. And after all of this optimization, we had 2 very good independent assays for these biomarkers. So you can see here on the left, the detection limits for the plDH portion of the assay were on the order of 5 parasites per microliter, which for plDH is actually a pretty good detection limit. In fact, if you compare it to commercially available ELISA kits, you can see that this assay outperforms the commercially available kit by an order of magnitude. Importantly, our assay took just 30 minutes for detecting plDH. And as you can see by the proximity of the lines here in our small-scale validation study, we had very good intra and inter-assay variability, so it was highly reproducible. We also had a similarly well-performing assay for HRP2, which had a detection limit on the order of 0.2 parasites per microliter, so again, very sensitive for protein-based detection. The total incubation time was just 20 minutes, and again, it was highly reproducible. So at this point, we had 2 independent assays that could be performed in a fraction of the time that it takes to do a traditional ELISA, and both assays outperformed commercially available kits by an order of magnitude. And so next, we wanted to put the assay the 2 assays together into a multiplexed format. So we have our magnetic beads that are functionalized with plDH-specific antibodies, as well as magnetic beads functionalized with HRP2-specific antibodies. And we also have our 2 detection antibodies for each biomarker. And what's important here is that these antibodies have distinct detection enzymes. So for plDH, we used alkaline phosphatase, and for HRP2, we used horseradish peroxidase. And in order to carry out this assay, we add all 4 of these components directly to a lysed whole blood sample. We allow the sandwiches to form directly on the surface of the magnetic particles, and we process the beads simultaneously using an external handheld magnet. So I want to pause here to mention that when I say multiplex detection, in this case, what I'm really doing is I'm doing multiplexed capture and processing followed by sequential detection. So once we've captured our biomarkers on the magnetic particles, we then incubate them in the detection enzyme substrate for plDH before then detecting HRP2 in the detection enzyme for horseradish peroxidase. And so the first step for multiplexing this sort of assay is to determine which detection step comes first. And so the best way to figure that out is to test it both ways. So as you can see here on the right, when we performed the plDH detection second, we saw substantially reduced performance in that assay, whereas for HRP2, we saw no difference between when HRP2 was detected first or detected second. And we think this happened because the detection enzyme substrate for horseradish peroxidase is acidic. It's a citric acid buffer. And we think that deactivated our alkaline phosphatase enzyme. And so we tested this hypothesis by performing an independent plDH on-beat ELISA with an additional incubation step in the same buffer that's used for horseradish peroxidase turnover. And as you can see, the performance of the assay was substantially reduced, confirming our hypothesis and leading us to detect plDH first, followed by HRP2. And so these are the results of a small-scale validation study that we did on the multiplex assay. We had 2 users performing the assay in sextuplicate over 5 days. And as you can see, our very good limits of detection were maintained, and we had good intra and inter-assay variability, and as well as very low inter-user variability, as evidenced by the proximity of the gray and black lines here on these graphs. We also wanted to see whether a novice could perform this protocol. So we gave the protocol to someone in our lab who had never done this before and gave her 4 unknown samples. And you can see her results are these red points here. And you can see they overlap very nicely with the results from the expert users, demonstrating that this assay is simple enough for a novice user to perform. And so just to summarize what we know about the assay so far, we're able to detect both biomarkers from a single whole blood sample. We know that this assay outperforms both commercial ELISA kits for both biomarkers by an order of magnitude. It's simple enough for a novice user to achieve accurate results. And importantly, the total incubation time for the assay is just 35 minutes. So we're able to detect 2 biomarkers in just a fraction of the time it would normally take to perform an ELISA for 1 biomarker. Importantly, unlike many other multiplex platforms, This assay requires no special instrumentation beyond what is needed to perform a traditional well plate ELISA. So you don't need a fluorescence microscope or a flow cytometer, for example. And because of this, we thought that this assay could be very useful in, for example, a regional lab in a lower middle income country. And so we wanted to try that out, and we took the developed multiplex assay to Macha, Zambia. However, the assay that was developed for use in lice whole blood samples. But the samples that were available to us in Macha were dried blood spots. And dried blood spots, as many of you know, are the most common sample format for large-scale studies, and they're most frequently used to detect genetic material. They're very advantageous because they allow for stable storage, reduced biohazard risk, and they're typically very cost-effective in terms of collection and storage compared to whole blood samples. There is a disadvantage of dried blood spots, which is in order to measure anything from a dried blood spot, you have to extract the material off of a paper. And to do that, there's an inherent dilution during the extraction step, which can ultimately decrease the assay sensitivity. And when I started to adapt the OBE ELISA for use with dried blood spots, I noticed that in the extract there are lots of small pieces of paper and fiber that cause biomagnetic particles to aggregate. And this aggregation resulted in high variability and high background signal, which really even further decreased the sensitivity of the assay. And remember, we want a high sensitivity assay. So my solution for this, I knew that we needed to filter these samples and get rid of those small fibers. And I wanted to do so in a way that could easily be done in Macha, Zambia, and also be done quickly, and not every individual sample needed to be filtered independently. And so I came up with this filter where using small segments of nylon fabric that actually came from pantyhose that you can buy at your local Walmart. And so we had, what we did was we cut the bottoms off of a 96-well PCR plate, and then we secured the nylon material over the bottoms and embedded that directly into a 96-well plate. So the DBS extracts could be pipetted directly through the nylon fabric and therefore filtered. And this filtering greatly improved our reproducibility. As you can see here, the error after filtering was significantly reduced compared to before filtering. And very importantly, it drastically reduced our background signals that we were getting for both biomarkers, which improved the sensitivity of the dried blood spot-based assay. And so the samples that we had access to were actually collected in Enchilenge, Zambia, which is a high transmission area during the rainy season from 2014 to 2015. The samples were provided by 15 children who were admitted to the hospital with acute malaria and treated with an artemisinin combination therapy. For each of these 15 patients, 15 samples were collected at different time points over 35 to 52 days, and parasitemia was measured at the time of collection using microscopy. So because of the nature of these samples, our goal was to use the multiplex dried blood spot-based OBE ELISA to measure plDH and HRP2 clearance patterns over time compared to parasite clearance. So what we did was we measured the concentrations of plDH and HRP2 at each time point. We then normalized those measurements to their highest value for each patient. And so here on the left, you can see the clearance, the parasite clearance pattern as determined by microscopy over time. As you can see, plDH follows closely with parasite clearance, whereas HRP2 remained in circulation long after parasites were cleared. In fact, by the last time point of the study, which was supposed to be 35 days, but many patients were there even longer for up to 52 days, at the last time point, 87% of patients still had HRP2 in circulation that we could detect using the OBE ELISA. And so if we look at the clearance, times for all 15 patients, as I mentioned before, you can see that the median clearance time for plDH was very similar as for parasites. However, we did see a few outliers, which we think may have been due to reinfection or recrudescent infection. HRP2, on the other hand, for most of our patients, we couldn't really define a clearance time because many of them still had HRP2 in their system at the last time point. So most of these data points are just the last time point in our study. And so these patterns not only match what has been seen in the literature so far, but this persistence of HRP2 for anywhere from 35 to 52 days are some of the longest studies that have been done. And we think that we could detect HRP2 for so long because of the sensitivity of our assay. And so just to summarize this first part about malaria, we developed a very sensitive tool for measuring plDH and HRP2 concentrations in samples with low parasitemia. And we accomplished all the goals we set out with. So we had single parasite per microliter detection limits. We could perform the assay very quickly, in just 35 minutes, compared to a traditional ELISA, which can take much longer. We had a high information output, detecting both biomarkers from a single sample, and had highly reproducible results. We then adapted this on-beat ELISA for use with dried blood spots, which again is the most common sample format for large-scale epidemiological studies. And we think that adapting it to dried blood spots is important because protein detection on dried blood spots is not very common in these large-scale studies, perhaps because ELISAs take so long to do. So we've reduced that time burden for protein detection. And then finally, we applied this assay to patient samples for 15 patients and measured biomarker clearance patterns over time. So now I'm going to completely switch gears to talk about schistosomiasis, which is a freshwater parasitic disease that's caused by Schistosoma trematose. And it's prevalent in many areas of the world which lack water sanitation infrastructure. There are 5 species of schistosomiasis known to infect humans, shown here on the left, with the 3 most prevalent being Schistosoma mannsoni, Schistosoma hematobium, and Schistosoma japonica. I won't go into all of the details of the life cycle, but I'm going to point out a few really interesting features. So humans are infected with schistosomiasis when they come into contact with contaminated freshwater source. And larval forms of the parasites penetrate the skin to infect humans. These parasites then develop into adult worms and eventually pair off and migrate to blood vessels associated with the intestines and the bladder. There, the females lay eggs, some of which are shed in the feces or urine, and when those come into contact with freshwater reservoirs, they can then infect the intermediate snail host to continue the transmission cycle. Schistosomiasis is considered to be the second most socioeconomically devastating parasitic disease after malaria, and the burden of schistosomiasis is immense. Approximately 700 million people are at risk of the disease, which is estimated to cause 200,000 deaths annually. And so, like malaria, having good diagnostics for schistosomiasis is very important in many of the same contexts. So for case management, determining whether someone's symptoms are coming from a schistosomiasis infection can help inform treatment. For intervention management, having good diagnostics allows us to determine whether an intervention such as mass drug administration or molluscicide treatment of a local body of water has reduced transmission within a particular population. And for surveillance, having good diagnostics is especially important because it allows us to perform large-scale studies which can then identify bodies of water which are sources of schistosomiasis transmission. And again, we also need to be, if we are interested in eliminating the disease, we need to be able to detect low-density infections so we can then treat those individuals. And so what are the tools that we have now for detecting schistosomiasis in the field? Well, the gold standard is light microscopy, which simply relies on the detection of schistosome eggs in stool or urine, depending on the species. And light microscopy is advantageous because it's affordable and it's considered to be quantitative of disease burden. So more eggs is generally considered to track with a higher density infection. However, there are several disadvantages to light microscopy. It's extremely labor intensive, and while egg counts are thought to track with infection intensity, they can vary dramatically between day to day. It also lacks sensitivity for detecting those low-density infections. So recently, one rapid diagnostic test has been developed for schistosomiasis detection. And it detects a proteoglycan waste product that the worm produces in its gut, and that waste product is present in human urine. So this RDT is advantageous because it's very affordable and easy to perform, and it takes very little time. But there are several disadvantages to this one particular RDT. First is that it only detects Schistosoma mansoni, so it can only detect one species of schistosomiasis out of the 5. It's only semi-quantitative, and perhaps its worst disadvantage is that it lacks specificity. So it's hard to see on this projector, but the field has come up with a term for when the RDTs are only slightly positive, and they call it a trace line, which is there, I promise. And these trace lines can actually show up in cases of hematuria, which can arise from schistosoma infections or many other infections or health issues. Additionally, these trace lines show up very often for pregnant women. So taking this test isn't necessarily good in terms of specificity. And so to mitigate these issues, our collaborators at Leiden University in the Netherlands have been developing a rapid diagnostic test that detects schistosoma biomarker called circulating anodic antigen, or CAA. And the reason why it's called circulating anodic antigen is that it's highly negatively charged at physiologic pH. And this is because it has these repeated disaccharide epitopes, which all have negative charges. It's produced in the gut of all species of schistosoma, and it's detectable in both serum and urine of infected individuals, regardless of the species they're infected with. And based on non-human primate studies, it's been estimated that a single worm pair of schistosomes produces 3 picograms per mil of CAA in whole blood. We don't know what that corresponds to in urine, but we know that it's the levels will be significantly lower in urine. And so, as I mentioned, our collaborators have been developing a lateral flow assay for detection of this biomarker. And it has a lot of really interesting features. It takes just a 10 microliter urine sample, and the detection limits of the assay are 10 picograms per milliliter, which is actually pretty good when you consider that a worm pair produces 3 picograms per milliliter in whole blood. The sensitivity is, one of the reasons why this assay is so sensitive is because of the unique detection probes that are used, which are upconverting phosphor nanoparticles. And these nanoparticles absorb low-energy infrared light and emit higher-energy green light. This upconversion is not very common in nature, and so we don't see any interference or background from biological or assay components. But one of the primary disadvantages of this lateral flow assay is that it can only be performed in the lab, which is obviously not ideal if you want a field test. So these urine samples actually have to go through several sample preparation steps, which require many centrifugation steps, including cooled centrifugation, which is not ideal. And so our goal was to not only improve the sensitivity of the lateral flow assay, but also make it more field-friendly. And I approached this problem by separating the diagnostic into its component parts and specifically focusing on how can we isolate and concentrate our biomarker of interest from the variable urine sample matrix. And so the most intuitive way to increase the sensitivity of an assay is to deliver more biomarker to the test. However, lateral flow assays are limited in the sample volumes that they can hold. So we decided to use a similar approach to what I just talked about and use magnetic beads to concentrate CAA from large volume urine samples. And we chose to do this for many of the same reasons we use magnetic beads in the on-beat ELISA. They afford us a high surface area and can be actively mixed within the large volume sample, which results in rapid capture times, and they're easily manipulated with a handheld magnet. So the way this would work is magnetic beads are added to a large volume urine sample. In this case, large volume is just 2 milliliters, but we can imagine going much higher. We then use an external handheld magnet to remove the supernatant and add a small volume of elution buffer, which removes the CAA from the particles and can then be added directly to the lateral flow assay. And so the strategy for capturing CAA on the surface of the magnetic particles that we used was by functionalizing these particles with polyamidoamine dendrimers, which are really interesting radially symmetric polymers, which have a lot of terminal amines. And all of these terminal amines are highly positive at physiologic pH. And these amines are important for 2 reasons. First is they're very easy to do chemistry with, so we can easily couple these polymers to magnetic particles. Second, because they're positively charged, they can interact with our biomarker CAA, which is very negatively charged, based on simple electrostatic interactions, which means that we can then elute, once we capture CAA using these electrostatic interactions, we can then elute the biomarker off of the magnetic beads simply by using a high salt running buffer, which disrupts this interaction by forming a double layer on the surface of the magnetic particles. And so I don't have time to show you all of the optimization data, and this is the final protocol that I came up with for concentrating CAA from these urine samples. And you'll notice immediately that it doesn't exactly look field-friendly, and I will tell you, I'll be the first person to tell you this is, in its current form, is not field-friendly, but we did eliminate all need for laboratory equipment, and it's the first step toward a point-of-care concentration method. And so the way this works is we add a small volume of trichloroacetic acid to our urine sample, which we then filter through a syringe filter. I am a little curious if that pantyhose filtering method could also work here as well. We then neutralize our sample before adding beads, and they just incubate within the sample for 5 minutes. We use an external handheld magnet to discard the supernatant and add our running buffer for just 5 minutes before adding that running buffer directly to the lateral flow assay. And so this is a graph of the performance of the original lateral flow assay as developed by our collaborators in Leiden University. And you can see that, as predicted, the detection limit was about 10 picograms per ml. When we performed our concentration method in mock clinical samples, so urine spiked with CAA, you can see that we improved the detection limit all the way down to 0.05 picograms per ml CAA, which is remarkably the full 200-fold enhancement limit that we could possibly expect. So this is really exciting. We've significantly improved the sensitivity of the assay. So we decided to apply this to patient samples. So far we had just been working in mock samples. And the patient samples that we were provided were collected in Egypt in 1989 to 1990. So they were about as old as I am, and stored in the freezer until I analyzed them. When I started to work with them, I noticed that we weren't capturing all of the CAA that we wanted to out of these samples. And we think this is because the patient samples had a much higher specific gravity compared to our non-endemic control urine. And we hypothesized that that means there's a much higher salt concentration in those urine samples, which interrupted the electrostatic interactions between our polymer and CAA. But I found that if we just diluted the samples tenfold, we could still get 20-fold enhancement in the limit of detection of the assay. And so I went ahead and did that for these 15 patient samples. And as you can see, we had an average signal enhancement of 22-fold, which is about the same as the full theoretical limit. And if we look at the 15 individual samples, these are the results using the original protocol developed by the Leiden group. And we looked at 5 samples that were initially negative by that protocol, 4 samples that were borderline, so really close to the detection limit, and then 6 samples that were definitively positive. And when we performed our bead-based concentration method, you can see that we found 2 infections that were positive for CAA that otherwise would have been missed by the original protocol. We also were able to definitively decide that the 4 borderline infections were positive using our bead-based concentration method. And so one of the things that you want to be sure when you develop a method like this and you start seeing infections where previously you weren't, is you want to validate them against an established laboratory-based method. And so we validated our results against a spin filter concentration method. And as you can see here, our results matched those from the spin filters, confirming that we had found these true infections that otherwise would have been missed. And so to summarize my schistosomiasis work, PAMAM functionalized magnetic beads were successfully used to concentrate CAA from large volume urine samples. And in mock clinical samples, we were able to achieve that 200-fold theoretical enhancement. It also enabled definitive detection of CAA in samples that would have otherwise been missed. And I'll be the first to say that the method that I came up with can't be used in the field. However, we're really excited about this PAMAM-based capture strategy, and so we're moving toward a fully integrated paper device that could reduce user steps for optimal field performance. And so the idea here is, instead of putting the dendrimers on the magnetic particles, we would put them on a cellulose-based capture membrane using really simple chemistry. We could then add a large volume urine sample to a test, which would be absorbed by absorbent pads, before sliding this capture membrane over top of a conjugate pad of a lateral flow assay, before adding the elution buffer to detect the CAA downstream. So we're really excited about this design because it's really just one extra step for the user of sliding the membrane from one side of the test to the other. And we think that this could have really great results in the field for concentrating CAA. And so to conclude my talk, I discussed the development of the Multiplex on-bead ELISA, which is a tool capable of measuring 2 malarial biomarkers at concentrations in the asymptomatic regime. We then applied this Multiplex on-bead ELISA to dried blood spot patient samples and characterized plDH and HRP2 clearance patterns. And finally, I developed a sample preparation method that enhances the detection of schistosomiasis up to 200-fold and enabled detection of low-burden infections that otherwise would have been missed. And I just want to emphasize that the way I approached all of these problems was by breaking the diagnostic down into each of its component parts and optimizing and evaluating each part to put them back together into an integrated assay. And so with that, there are many people to thank for this work. First and foremost, my Ph.D. advisor and my committee members who provided a lot of guidance along the way. I especially need to thank the collaborators who made all of the work with patient samples possible, especially Dr. Phil Tuma in Macha, Zambia, as well as Dr. Hobart Van Dam and Paul Korstens in the Netherlands, who generously hosted me in their lab for 3 months. Of course, the members of the Wright Lab all provided invaluable advice and guidance through problem solving. And finally, I need to acknowledge all of the funding that made this work possible. So thank you all again for hosting me here. I've had a really great time so far, and I'd be happy to take any questions that you have.
How do you actually visualize the results of the malaria test? You need a reader or by eye?
So we used a typical UV-Vis plate reader, but you can see them by eye. We haven't studied how sensitive it would be by eye, but we quantitate using the plate reader.
And do you envision being able to do single, like if a fever case comes in, you need to diagnose that one, or do you see it more as a surveillance tool? You do batches.
We see it more as a tool, a research tool that can be done in batches. I think in terms of it, obviously the OBE ELISA would be very hard to perform at a point of care setting. It is definitely a good tool for the laboratory, and that's why we think it would be good in terms of research and surveillance.
Well, Christine, great talk. Thank you. I'm curious about this life of HRP2.
Mm-hmm.
You're seeing positive in 40 days, 50 days.
Mm-hmm.
So this is also from asymptomatic patients? These are clinical patients.
Those are clinical patients.
That's really interesting because for the RDT that we use in the field every day, we know that it's positive for about 2 weeks, I think, right? It's 10 to, it's actually PCR is longer.
PCR is longer.
PCR is longer. Shorter. Yeah, PCR is shorter. So I wonder what that means, right?
Yeah.
What does it mean, and what's the implication? Is it good or bad?
Well, it depends on what your goal is.
Right.
If you're looking for active infections, it means your test is much less specific because you're potentially identifying recently resolved infections, where recently can be anywhere from a few weeks to, I guess, 35 to 52 days.
How long did you look at, by the way?
The last time point was supposed to be 35 days. Some patients didn't come in until after that.
Accidentally.
Yeah, exactly.
So you talked about the clearance of LDH and HRP, like how long it takes them to clear, but do you know, or is there information on after the onset of an infection, how long it takes those protein levels to build to a detectable level?
That's a really good question, and I didn't do that study. That would probably have to be done in a controlled human infection study, and I'm not sure. The data, I'm not sure that anyone has done that specific study for those two biomarkers. But I do know that HRP2 accumulates over time and remains in circulation. So in general, HRP2 is much higher concentration compared to p.DH, which is generally thought to track with parasitemia.
There's no data on that from challenge studies?
I haven't seen it. It would be an interesting study. Yeah.
Well, that's why we have to get challenge studies set up.
Right.
Do you know how much the malaria test costs?
The OBE ELISA?
Yeah.
It's approximately the same as a plate ELISA, so we haven't really improved it because it still uses about the same amount of antibodies. I don't know the number off of the top of my head, but we haven't improved the cost, just the time and sensitivity.
The first slide you showed was just some of the basic stats of malaria, the total infections and the total deaths. Given that you are now an expert in diagnosis, is that an underestimate or an overestimate?
It's an underestimate.
It's an underestimate.
I think even the World Health Organization admits that those numbers are probably an underestimate, not only due to diagnostics, but also reporting.
I thought it was overdiagnosed. Is that just clinically then, but that a lot of the infections are asymptomatic and so they're never going to clinic?
I think so.
But it's also probably overdiagnosed in the clinical sense, and that people get a lot of treatment when they don't have malaria.
Right.
But the WHO numbers are generally going to be rapid diagnostic test positive cases that are kind of reported up through the public health systems, which in turn underestimate the actual number of. So they're kind of, going wrong in both directions.
Right, exactly. Perhaps they even each other out.
Yes.
You mentioned the 5 species of malaria that could potentially be part of the test. And which considerations did you have for choosing specific biomarkers?
So in terms of looking at Plasmodium falciparum and Vivax for p.DH, we chose those 2 because they're the most prevalent species around the world. But it would be interesting to do a similar antibody study for all 5 species. That would be a really interesting question to ask. See if we can come up with a true, because even there are tests that, like rapid diagnostic tests that are advertised as pan tests, but they don't work the same for every species of malaria. So it would be interesting to try to look for something that would work.
I'm just curious about your experience in the Zambia, in the lab in Zambia. Do you think it would take a lot of training to scale this up so that it can be?
I don't think it would take too much training with the OMPs ELISA, you mean?
Right.
Yeah, I think, especially, when I was in Zambia, all of the lab technicians who were there were very skilled. I was really impressed, and I think they could learn this in a day.
I've got one more question. So it's kind of related to Ryan's question about kind of the trajectory of parasitemia on infection. Is there any way in the lab to tell, so 2 people might at the cross at a point in time the same parasitemia load, but one ends up becoming infected or symptomatic but just hasn't gotten there yet, and one might stay asymptomatic forever. Is there a way in the lab to sort of predict what that trajectory is going to look like?
That would be really great.
That's why this is so important.
Yeah. I do know that HRP2 concentration is a predictor of severe malaria, so getting cerebral malaria. Having high HRP2 concentrations can predict that, but I don't think we have a tool for knowing whether anything will stay asymptomatic.
I think we would need a protein microarray to measure antibody responses to all the different variants of key antigens and full genome sequencing of the infecting parasite, and see where there's a mismatch, and where they, the ones that are lacking antibodies to the parasites they're infected with would be the ones we would predict would go on to get sick. But it's a little bit out of reach for
And there's also a whole aspect of, the difference between symptomatic and asymptomatic parasites. So we talked about the truly asymptomatic parasites, whatever we know much about malaria parasites is coming from the clinical, acute malaria. Right? So we talk about metabolic, differences there may be in the asymptomatic parasites.
Right.
Young people will sort of remember.
So you showed that curve for the clearance of the clinical cases, and that was with your diagnostics, which were really, and I may have missed this, but were there comparison curves as to, presumably they're going to be a lot longer because you're detecting it for longer because the test is more sensitive. Are those curves, how are they different for the less sensitive tests that people are using?
So unfortunately, we didn't have enough sample to do multiple biomarker concentrations. So all that we could compare our biomarker concentrations to was the parasitemia determined by microscopy. But as I mentioned, in general, the clearance times that we saw for p.DH matched previous literature studies, and HRP2 these are some of the longest measurements for HRP2. So I don't have the comparison already.
So it sounds like it may not be necessarily clinically meaningful at that length of time for
HRP2.
I can't imprint the first one, the p.DH. Is
Yes,
that?
p.DH,
Yeah.
yes.
Okay.
Yes, so yes, you're right. The assay sensitivity of the p.DH portion of the test was was decreased when we applied it to the dried blood spots compared to the whole blood samples. And so, so the data that we got was matched, but didn't provide us any new information. Whereas the HRP2 basically maintained its very high sensitivity when we applied it to dried blood spots. So we did see that extra long persistence. Yes?
On co-infection, so could a diagnostic like yours be useful for detecting when someone has multiple infections, say malaria at the same time? And is that valuable to be able to detect?
Yeah, that would be very valuable. The assay in its current format wouldn't be able to distinguish between a co-infection and a falciparum infection.
Because since it works for everything, it should know. Is there any way to...
Yeah, so you can, just like all of those antibody pairs have different performances for each species of p.DH, you could imagine, if you could imagine choosing an antibody pair that was specific for one species of p.DH to get that information out.
Thank you all so much for coming, and thank you, Christine.